So good afternoon, everyone. My name is Alex Bohemsky, and I'm a high school student at Hamden Hall Country Day School in Hamden, Connecticut. And I work with Dr. Risparo at the chemistry lab there. And thank you for the opportunity to present research at this year's symposium. So sorolins are compounds that occur naturally in the plants Sorrelia coralifolia and also Animagus, found in India and Egypt, respectively. And they act as fungicides in these plants. And chemically speaking, though, the sorolin molecule is tricyclic, and it forms part of the class of linear fluorocoumarins. And the linearity of the molecule and actual flatness of it allows it to both absorb energy very well and intercalate in various biological constituents like DNA or proteins. And in essence, sorolins have been used since ancient times in actually Ayurvedic medicine. They used to apply poultices of sorolin plants to affected areas of patient's skin and allow the patient to stay in the sun. And actually, they were practicing a crude form of PUVA therapy, sorolin plus ultraviolet A therapy. And you'll also find applications in dermatology and viral inactivation. And our actual experiments focus on 8-methoxysorolin, or 8-MOP, and 4'-aminomethyl trimethylsorolin, or AMT. But in 1955, Musaio, a group in Italy, characterized the reaction of sorolins with nucleic acids. And he showed that sorolins can migrate and intercalate at selective sites in the DNA sequence, particularly adenine and thymine. And following that, they are also excited by ultraviolet photons. And upon the absorption of one, the sorolin can form a monoadduct at either the 3'-4 site of the molecule or 4'5'. And provided it's a 4'5' monoadduct, a second photon will cause crosslink formation. And all those adducts and crosslinks hinder DNA replication and cause cell death. But recently, sorolins have emerged as anti-cancer agents, and as we know, cancer is characterized by increased DNA replication, uncontrolled cell division, but most tumors are located in the internal organs, as shown on this person here. But the problem, though, is that ultraviolet to activate the sorolins can't penetrate deep enough to these areas, and also adduct detection and the assessment of these sorolins is time-consuming, and it's cumbersome almost. But the solution that has been developed is the use of synthetic tags for in situ photon delivery and also a more targeted drug delivery to minimize adverse effects to otherwise healthy parts of the human body. Now, the key to all of this, our collaborators at Duke University have pioneered work using nanoscale simulators. And these are usually made out of transition metal compounds like yttrium oxide. And they're actually used to convert high-energy X-rays that can penetrate deep into where tumors are located and convert that energy into UV radiation that can activate photoactivable drugs. And this is plasmonics-enhanced photospectral therapy. And plasmonics is the field where light can be used to store information. And this also helps with the more targeted delivery. And the most effective configuration they found is by tagging this nanosimulator with the HIV-1 TAT peptide, which is cell targeting and nuclear penetrating, to get the drug directly to the nucleus and the DNA where it can take its effect. And then tagging the sorolin subsequently on top. And this is a figure from their paper where they show the spectral overlap area between the absorption spectrum of the sorolin and the emission of the yttrium oxide. So you're creating photons where you can excite the molecule and generate that cell death. So our objective, however, was to optimize modalities for activating and detecting the DNA incorporation of synthetic sorolins that are being used as anti-cancer agents. Because sorolins, when you tag the nanosimulator and the HIV-1 peptide, you actually impact its biological characteristics substantially, and binding constant is subsequently altered. So our study design then was to optimize adduct detection by using a new mass spectrometric technique called CELVI-TOF, and also by looking to optimize in situ photon delivery by using UVB instead of UVA. So what is CELVI-TOF? It stands for Surface Enhanced Laser Desorption Ionization, Time of Flight Mass Spectrometry. And the keywords there are surface enhanced, because each chipperite is manufactured with a different surface to attract a different type of analyte, whether it be hydrophobic, positively charged, or negatively charged. There's a whole array of chips that can be purchased. And that's how you can separate complex samples with minimal handling. So whatever you want to analyze is attracted to this surface. So it combines, therefore, chromatography and mass spectrometry, and the key thing here is that it's rapid. So whereas data could have taken 2 days to obtain with HPLC, you can obtain it in 1 hour with this technique. And we were one of the only groups that decided to use non-protein analytes with this technology. And therefore, we decided to use a Q10 array, which is positively charged, to attract the negatively charged DNA phosphates. So this is how you prepare a CELVI. A complex sample is added, incubated for about 1 hour, and then subsequently washed. and then a matrix is added as it would be done with MALDI. And we found the best one is actually hydroxypicolinic acid and also ammonium citrate to reduce matrix saturation on the detector. And this actually proved to be very effective in the long run, as I'll show later. So why did we choose UVB instead of UVA? So usually you irradiate at 365 nanometers around here, but we wanted to decide to use 300 nanometers, which could actually generate adducts in a more efficient manner due to the higher absorption. And we also hypothesized that if we were to work with both 8-methoxysorlin, which is the clinically approved drug, and AMT, that the effect would be more pronounced with 8-methoxysorlin, because the ratio is about 4 to 1 here, and as for AMT, it's lower. So that's what we hypothesized. So now for our results. This is purely a theoretical calculation we did, showing how binding constant increases intercalated molecules, and we synthesized an oligonucleotide, 10-mer, alternating adenine and thymines. And it was antiparallel, so it annealed on itself. And this is where we detect with CELDI, UVB adducts. So these are typical CELDI tracings we look at, where we see the intact oligonucleotide at 3,025 Daltons, and also the intact oligonucleotide plus the AMT molecule, which the peak develops subsequently with increasing irradiation time. And we show that it's quantitative as well. It increases with time. So that result showed us that CELDI could detect and quantify. Now this is where we compare UVA and UVB activation of 8-methoxysorlin. And just as we expected, UVB shows a significant difference than UVA. And we also showed this as well when we wanted to talk about how UVB would be the better activating radiation, because again, with sorolins that have such low binding constants, when you're dealing with all these nanosimulators and peptides, you need to be able to activate the drug very efficiently. But when we expanded these results to AMT, we saw that the difference was not as pronounced as it was for 8-MOP, and in fact, although there appears to be somewhat of a difference, it wasn't enough to reach statistical significance. And with that, we can offer a few explanations. We can say that AMT, with a binding constant of approximately 150,000, has such a high constant that it is intercalating at so many sites that the type of irradiation isn't as significant as it would be for 8-methoxysorlin. So our future directions then are to characterize adduct types. Are monoadducts and crosslinks equal in the therapeutic setting? And also we want to assess the wavelength dependence of adduct formation. How is it that some wavelengths can generate monoadducts while others can generate a preponderance of crosslinks? And both of these techniques would actually require HPLC, but we hope also to develop an optimized CELDI protocol for the above objectives, probably involving a digestion step to differentiate between monoadducts and crosslinks. And the conclusions we can offer for sure, though, are that CELDI-TOF is able to directly detect and quantify sorolin photoadducts, and adduct formation varies with the type of sorolin and with the type of UV irradiation, as we've shown with the difference between 8-MOP and AMT, and also that UVB can be used to overcome sorolin's low binding constant and quantum yield to increase its adduct formation. And this is especially applicable in this developing field of synthetic sorolins with sorolin functionalized nanosimulators. So, in my acknowledgments, I'd like to thank the Keck facility at Yale University for helping us synthesize our oligonucleotide, and the Brash Laboratory at Yale for letting us use their radiometer to measure the output of our sources, and definitely Immunolite for giving me my first $500 grant, and also Hammond Hall Country Day School where I attend. So, thank you so much.
Questions for Alex? It's very exciting. So how do you differentiate between, so how do you administer the light for the sorolin cross-linking, and then how do you differentiate that from when your analytes are laying on your array and you're light desorbing them, right? So
yeah.
You're also using high energy there too. So,
yep. So actually, in the therapeutic setting, I think with X-rays, how do they do that? They would deliver the drug directly to the tumor and then allow the patient to be irradiated with X-rays, probably in a flash photolysis setting with X-rays, and that would trigger the nanosimulator, and that would then trigger adduct formation and cell death in the tumor. But with our CELDI arrays, we irradiate separately and then apply the sample to there, and I hope that explains what you're asking. Makes sense.
Yeah, I think so.
Yeah.
Any other questions for Alex? All right, thank you, sir.
Thanks a lot.